Hello, my name is Carol Leibel, and today we're going to examine those little biological catalysts we know as enzymes. If you were to mix 2 moles of hydrogen gas with 2 moles of oxygen gas, nothing happens. But if you were to add a spark to the container, you could have an explosion. Kaboom! It turns out that that spark adds a little bit of energy to the mixture, and that little bit of energy is used to break the bond that's holding the 2 hydrogen atoms together and to break the bond holding the oxygen atoms together. So for an instant, the H2 becomes 2 atoms of hydrogen and the O2 becomes 2 atoms of oxygen. And that's going to free up those electrons that were tied up in the covalent bonds so that now they can form a chemical bond in the formation of water. This turns out to be an exergonic reaction. Delta G is a minus 58 kilocalories per mole. I've mentioned this before in an earlier PowerPoint, but the Hindenburg is an example of what happens when hydrogen gas with a spark combines with oxygen to form water. There was a terrible, terrible explosion. Now, that spark is called the energy of activation. And as we've mentioned, during a chemical reaction, bonds have to be broken and bonds have to be remade. So in this case here, those bonds are broken. And then now down here, these bonds are made. And the changes between the energy of the reactants versus the energy of the products is called delta G. And when you take the energy of the products minus the energy of the reactants, and it's a negative delta G, then we have an exergonic reaction. Now, enzymes are biological catalysts that increase this reaction rate. Characteristics of enzymes are they're made up of proteins. However, recently they have found out that there are some enzymes that are actually made of RNA, which is quite amazing to me. They're very specific and only work with a certain set of reactants or substrates that fit on their active site. So here we have an enzyme, and somewhere there's going to be an active site in which a substrate can fit. So structure is all important to making an enzyme work. Enzymes can be used over and over again. When a substrate attaches to the active site, you'll notice that the enzyme is going to change shape. That's called the induced fit. And when it changes shape, that change in shape will help the chemical reaction occur. Here is an example of our picture of an enzyme called ribonuclease. Let's go forward. Ribonuclease. And this particular enzyme has the ability to break down nucleic acids so that the nucleotides can be recycled and used over and over again. The purple part that you see right here, that is the enzyme, and then the green is actually the nucleic acid that is being broken down by the enzyme. So how do enzymes work? Well, enzymes work by decreasing the energy of activation needed. They do not change the Gibbs free energy of the reaction. So here's the energy of the reactants. Here's the energy of the products. The difference in the two is delta G, or a change in their free energy. And you'll notice it doesn't matter if with the enzyme or without the enzyme, delta G stays the same. However, what you do notice is that that amount of energy right there is the energy of activation needed to make the chemical reaction work. So with an enzyme, what happens is that the energy of activation is actually lowered. Here's an example of an enzymatic reaction. You know that when you eat something like sugar, it has to be broken down. Sugar is a disaccharide. It's made out of glucose and fructose, and you cannot put that directly into your bloodstream. When you ingest sucrose, you have an enzyme called Sucrase, which breaks down sucrose. So what is going to happen? Here is the active site, and it allows sucrose to fit onto the active site of the sucrase enzyme. Water then is allowed to come into this chemical reaction, and now what happens is that we have a chemical reaction taking place in which now we have produced two monosaccharides, one being glucose and the other being fructose. These molecules are now small enough that we can put them into our bloodstream. Those two now are going to leave the active site, and so now this enzyme sucrase can be used again and react with another molecule of sucrose. So here's a little video that's going to show you what happens during an enzymatic reaction.
For any reaction to occur, even one that releases energy, some energy must be added to get the reaction going. This energy is needed to break bonds in the reactant molecules. The energy needed to start a chemical reaction is called the energy of activation, EA. This required energy input represents a barrier that prevents even energy-releasing reactions from occurring without some added energy. How does a living cell overcome the energy barrier so that its metabolic reactions can occur quickly and precisely? A special kind of protein called an enzyme is the answer. An enzyme serves as a biological catalyst, increasing the rate of a reaction without being changed into a different molecule. An enzyme does not add energy to a reaction. Instead, it speeds up a reaction by lowering the energy barrier. An enzyme is very selective. Its three-dimensional shape allows it to act only on specific molecules, referred to as the enzyme's substrates. As the substrates bind to the enzyme's active site, they are held in a position that facilitates the reaction. This takes less activation energy than the unaided reaction. Products form and are released. The enzyme emerges unchanged from the reaction. Because of the specific fit between enzyme and substrate, each enzyme can catalyze only
one specific reaction is the tail end of that little video. So now we're looking at an enzymatic reaction, and if you look at this graph, we're looking at the amount of, let's go back. We're looking at the amount of product being formed over time. And what we notice is that at the very beginning, the reaction rate with an enzyme happens very, very quickly. And then over time, the rate of the reaction starts to slow down. And the question is, why is it slowing down? In fact, at some point in time, it's going to flatline. The question is, why? Well, at the beginning, there is lots of substrate around. And so the likelihood that an enzyme and a substrate will make contact and the reaction takes place is quite high. But as the reaction proceeds, more and more product is formed. As more and more product is formed, then you decrease the likelihood that the enzyme is going to interact with a substrate. So the reaction rate is going to be fastest at the very beginning, and we call that the initial velocity. All right, so let's look at another situation. Let's look at another situation in which we repeat the experiment over and over again, but every time we repeat the experiment, we increase the concentration of the substrate. What we'll notice is that it increases, it begins to go faster, and then over time what happens is we can't, no matter how much substrate, we can't get the reaction to go any more quickly. The question is, why? Well, the reason for that is that all the enzymes are saturated. So the initial velocity, we can't get it to go any faster because every enzyme is already has already combined with the substrate. So we want to examine some factors that might affect how fast an enzymatic reaction will take place. So we're going to look at this graph, and at looking at this graph, we're looking at an enzyme that's a typical enzyme found in a human. And as the temperature increases, we start to see the reaction rate going faster and faster. And then we start to see the reaction rate plummet. So there's two parts to this graph. There's part A and part B. Now, what's the reason for part A? Why is it that as the temperature is increasing, we see the rate of the reaction also increasing? So just take a moment and think about it. Well, the obvious answer is that as the temperature is increasing, the molecules are moving more quickly, and that will increase the likelihood that the enzyme and a substrate will indeed connect and combine, and then the chemical reaction will take place. Let's look at part B. Part B, we now see that the rate of the reaction after we reach a certain optimum temperature plummets. Why is that the case? So take a second and think about that. This may take a little more thought, but if you remember that enzymes are proteins, proteins are held together in their secondary shape and some of their tertiary and quaternary shape by hydrogen bonds. And so as the temperature increases, you increase the likelihood that those hydrogen bonds are going to be disrupted. And remember, we said that the shape of the enzyme is so important to its function. So if you change the shape or the conformation of the protein or the enzyme, it's no longer going to function the way it's supposed to. And so after a certain temperature, it appears that the majority of the hydrogen bonds are just simply going to fall apart. And so the protein loses its shape. Now let's look at the red line. And it says that we have a thermophilic bacteria. These are bacteria that have been found in places like Hot Spring, Arkansas, in temperatures that approach boiling water. And when they extract those enzymes, what's interesting about those enzymes is that they function at a much higher temperature. And you can imagine that during the course of evolution, there was an advantage for those sorts of bacteria to have proteins that can tolerate that increase in heat without disrupting the hydrogen bonds. And so for thermophilic bacteria, the optimum temperature for the reaction rates of their enzymes is much, much higher than what you would find in humans. So temperature can indeed have an effect on the reaction rate of enzymes. All right, so here's another factor that can affect the reaction rate of enzymes, and that is pH. We have two different enzymes that we're looking at. One is pepsin, and the other is trypsin. Now, trypsin is found in the intestine, and it appears, looking at this graph, that the optimum pH in which this enzyme is going to work at the fastest rate is between 7 and 8, a little bit on the alkaline side. And that makes perfect sense, all right? But pepsin, this is an enzyme that helps break down proteins. However, it's secreted into your stomach. Now, what do you know about the stomach? In addition to having enzymes like pepsin that breaks down proteins, we know that there are acidic secretions or hydrochloric acid that's being secreted into the stomach. And so the pH of the gastric juices of the stomach is extremely low. So you have to have a protein that can function in a very acidic environment. And so it makes sense that we have an enzyme that natural selection has favored its ability to work in a very acidic environment. So if we turn around and we change the pH, so as the gastric juices move over to the small intestine, there is a change in the pH. And so what will happen, pepsin will not function in the small intestine. And so it makes sense that you have to secrete another enzyme that can help break down proteins into your small intestine, and that turns out to be trypsin. All right, so here's another factor that can affect the rate of an enzymatic reaction, and those are inhibitors. So let's suppose we're talking about, let's suppose we're talking about an enzyme, and here is a substrate that fits on its active site. And let's suppose we're talking about, sucrose, and this is sucrase. This is just hypothetically, all right? And along comes maltose. Now, maltose is very similar to sucrose. They're both disaccharides. Hypothetically, it would make sense that maltose could also attach to that active site. And so if that inhibitor were there, that substrate cannot attach. So having a competitive inhibitor around is going to lower the reaction rate just because a certain percent of the enzyme is going to be involved with the competitive inhibitor. Now, there are other times in which you have something that might attach to an enzyme that will cause the enzyme to change shape. But whatever is attaching to the enzyme does not attach to the active site. So in this case here, this is called a non-competitive inhibitor because it's not competing for the active site. But nonetheless, when it attaches to the enzyme, it causes the active site to change shape so that the substrate cannot attach. So a competitive inhibitor has the ability to attach to the active site. A non-competitive inhibitor attaches to the enzyme, but not at the active site, but causes the active site to actually change shape. So now we want to look at something called allosteric regulation. And here are two different examples of allosteric regulation. There is a site that will allow the attachment of an activator or an inhibitor. And the enzyme can take two forms, an inactive form or an active form. Now, if you have an activator, and an activator can attach to a particular region of the enzyme and stabilize the enzyme so that now substrates can attach, that's a form of positive allosteric regulation. But in another example, if instead of an activator, you have an inhibitor attached to the enzyme, and when the inhibitor attaches to the enzyme, it changes so that all the active sites are now cannot be reached by the substrate, then that's an example of negative allosteric regulation. So allosteric regulation is involved when you have an enzyme that has the ability to change into two different conformations or shapes, if you will. One is an active form and the other one is an inactive form. If you have a molecule that can attach to the allosteric site and cause it to be stable and allow the attachment of the substrates, that's called positive allosteric regulation. But if it's a molecule that does just the opposite and causes the enzyme to become inactive, then that's an example of negative allosteric regulation. And so taking into consideration things that can affect enzymes, we have to talk about feedback inhibition. So if you have a biochemical pathway, and in that biochemical pathway, there are a number of enzymes involved, and at the end, you have some sort of product, it makes sense that if that product is already in the cell, that you have a way to shut down that biochemical pathway. So in this case here is an end product, an amino acid called isoleucine. And here is the first enzyme in that biochemical pathway called threonine deaminase. And it has an active site, And so what happens, if there's lots of isoleucine around, it will attach to that particular enzyme and it will make it inactive. When it becomes inactive, it shuts down this biochemical pathway. If there's no isoleucine around, then that will be detached. The active site now becomes available, and now this biochemical pathway is going to be a go. We're going to go ahead and start making isoleucine. And so this is called feedback inhibition because the product has the ability to inhibit the biochemical pathway. And last but not least, we want to talk about cooperativity. Cooperativity is an example where you have a particular enzyme that has more than one active site. And when a substrate attaches to just one of the active sites, that will stabilize the enzyme so that now the other active sites are now available to interact with the substrate. So if you will, the substrate is cooperating with the enzyme. All right, so my name is Carol Leibel, and I want to say I hope this PowerPoint helped you understand enzymes and factors that can increase or decrease the rate of an enzymatic reaction. And I hope that you have a great day and enjoy the rest of your year in AP Biology.